Thank you, and good afternoon to everyone. I'm very happy that you come to my talk. First of all, thank you for the invitation to this wonderful program, and actually to give me the possibility to talk about the work that I have done in the past, and that I will continue in Guildford at the University of Surrey as soon as my lab and my group will be ready to go. So I will show you all the experiments that I have done, and I prepared this talk focusing on the open questions that I still have, because I think that somehow it's a little bit the spirit of this program. So I would. Okay. So all the projects I will show you in this during this presentation.
Just check what's in the chat because it's a Rhoda saying that she's sorry she couldn't make your talk.
Sorry, I did. It's already done. Okay, good. So I was saying that the project I will show you in this during this talk got inspired by the idea of reconstituting living systems. We had this project where actually the idea was of considering the cell, taking biological building blocks that we find in cells, and then having from living systems, and then assembling them in a way that then we get active structures, and then ideally through their self-organization they could mimic biological structures, and then accomplish biological functions, and then we could maybe having some model system that could help us to understand some physical processes that happens in biology. I will focus mainly on two structures, so cilia and flagella, and cytoskeleton, so the active network that we have in the cytoskeleton. Now, for both topics that I will show you today, the biological building blocks that I use are microtubules and motor proteins. Here I show only the kinesin, but actually in this talk, you will also see that I will use also the other family, the diving family of the motor proteins. And I don't think that in this audience I need to explain what microtubules and kinesin are. And actually, the thing is that these two components were assembled by following a bottom-up method, and then we could analyze their interactions in vitro. So for those of you that didn't know what cilia and flagella are, although we had a talk last week, but cilia and flagella are, I would say, hair-like structures that, in the case of cilia, they cover the cells, and that's why a tissue, forming a carpet that then synchronize in the way that then they can transport fluid in the case of cilia. And to have an example, if this transport fluid is not really, easy to imagine, just think on the movement of the mucus that we have in the lung. And this, the cilia basically help our body to expel what the body doesn't want to keep. In the case of the flagella, we have a longer structure that actually is used in the by organism to propel in a liquid environment. Now, this is our example from mammalian cells, but actually we have also other organisms, other eukaryotic cells that have these structures, like this one is an algae called Chlamydomonas, and with this, the flagella also in this case are used to propel in the environment. It is important to know that we have also other organisms, because then this one can be used as a model system. And the reason for this is because it doesn't matter if we are talking about the flagella of the Chlamydomonas or the cilia in the brain, they basically share the same structure. That is the basal body that link them to the cell, and then they protrude in the extracellular space, and they have this framework made of microtubules. And if we consider, like, let me say, the basic unit of this structure that is called axoneme, then we see two doublets of microtubules, and then the motors, in this case the dynein. They then, through their cycle of binding, working, and detaching, can just, let's say, give this beating movement that I show you in the movies before. And the bending is given by the fact that these microtubules are linked by special cross-linking proteins. When we began this project, we asked how simple can be the simplest system that is able to beat. And then we just tried with this very extremely simple system. One microtubule that is linked to the surface, and then you have some kinesin motors that move between the filament and the surface, that is treated in the way to reduce the absorption of the protein. By using this kind of system, you can see this movement, and then you see how these microtubules go from one side to the other side, and then buckles, and because of the elastic stress, then goes back in the straight position and begin the cycle again. Now, this you observe this continuous beating from one side to the other side. Actually, as long as you observe, I mean, you fix the visualization time. Our time was the maximum that we observed was 5 minutes, and then you do let's say the analysis to define a kind of period by defining just the straight position, and then the time that the structure needs to go from one side to the other side. And then we let's say we find the period, if we can talk about the period, that it was around 1 minute. Now the question is, at this point we didn't have the models, we didn't have labeled models, and that's why we didn't know how many models were let's say involved in this process, because we were not putting the models on the surface. We were mixing the microtubules with the models and then putting on the surface, and the concentration was very low. That's why much lower than the normal gliding assay. And then, I mean, as a kind of control experiment, we increase the concentration of the motors to a level like for example a gliding assay, and then we observe that the system is blocked, that actually you cannot get this kind of oscillation if you have too many motors. That's why this idea that you have all the surfaces covered with motors, and then when the microtubules move always attached to one motor doesn't really work. And
did you add something to make it sticky? Because obviously part of the microtubule was stuck, or was it just bad motors?
How the motors were attached to the
No, and obviously the microtubule sticks at some parts of its thing. So did you do that on purpose, or did it just happen to be sticky?
No, I mean, you do on purpose, because I mean you try to always to have a kind of clamped end.
Right.
Yeah, and this why I mean you have always this kind of, and actually this is you try to do in a specific way. It's not specific, but always you get it, because the surface is covered by peg brushes. But then if you treat in some position these peg brushes, you can let me say now in very open them, because these peg brushes are attached to the surface with polylysine. The polylysine is positive, the microtubule is negative, and then if they open in some position, they can clamp the microtubule, but not in the rest because of these brushes. Was this what you were asking?
Can I ask a question?
Yes.
It seems to me that what you are showing us is motors compressing the microtubules at back walls. Is that what we see? And is it the same thing as BT? It's not obvious that it's the same thing.
I mean, how the system works.
Previous movie that you showed, I have the impression that you are compressing it.
I mean, if you think you have, let's say that you have a model here, right?
Yes.
And then these models then walk in the toward the plus direction, but on the other side is linked to the surface, and in this way you have the buckling of the yes.
It seems like a buckling just in the middle. Compressed and is that the same thing as a BT?
No, absolutely not. No. I mean, no. I mean, this is again was a kind of a first attempt of having a bit of structure, but not similar to the no.
Is it possible to light up the diamonds so that you can?
So this is first of all, this was my mistake. For this experiment, we didn't use the diamond. We used a kinesin, just because always you have still the possibility to let the microtubule sliding. I mean, moving on the motors, and now you have this pattern.
You have the kinesin is the motor.
Yes, and you have to imagine this is a link to the surface, but actually we try to functionalize the surface in the way that this link is not too tight, but at least this is what I was hoping and my speculation. And then on the other side you have the microtubule, and then it walks in toward the plus, and then buckle this field. It's I would say a classical buckling filament that has one compound.
Can you visualize the kinesin?
No, this is the reason why in all this study we did all this control, because the level models were not available, and then we did this control experiment, and at the end we solved the problem with Ramin because Ramin then in the group of Ramin, they will show you. Yeah, let's say. And okay, and then to show that actually you really need a low concentration, if you put the half of the concentration of the motors, what you observe is that actually the filaments lose the attachment, and then they just get some movement, but then they cannot they don't have enough motors to have the repetition of this movement. But you can. You measure the tangent angle along the filament. In these chemographs, you see the pattern of the traveling wave along the filament that corresponds to the 4 cycles that I show you before. And if you compare it with the chemograph of a sperm, actually you somehow, this is a very helpful way to see when you use a slender body for representing a flagella, what you can keep for a flagellum, and what you lose actually in this approximation, let's say in this way. Exactly for the reason that we don't have level motors. The question was how many motors we need for having the deformations of these microtubules. And then actually, this is, I should say, I'm a particularly I have a very strong memory with this project, because it was the first project that I did with Ramin. Ramin arrived there just, I think, I don't know, 4 months before, so 4 months before, and we worked, and he helped me with this project. And the idea was that actually with a very easy model, they just took the deformations. And then they tried by using the lasting beam equation to find the force distribution that you need for having this the shape, the deformation of the shape that we observed during the experiment. And here, and you see here that actually below there is I don't know if you can see it is a gray line that is that represented the experiment, and then here this is the results of the model, and then if you take the force distribution and you decompose it, of course you are interested in the longitudinal component, and by considering that you are between 0 and 10 piconewton, then you can, by using the values in the literature about the Keynesian force, you can guess that for this kind of movement, 2 motors are enough to give you this deformation. And this was, let's say, a way how we solve this question that I told you at the beginning. How simple is the simplest system that is able to beat? But I show you this kind of let's say unit. And this is, the motors are not the Keynesian, but it's the Danae. And on the top of this, the motors are interacting with another microtubule, and not with the surface, as I show you, because it's not really absolutely not closer to the reality. For this reason, then we collaborate with Kazuhiro Oiwa. Is an expert in the field of axonymal dynein. That is exactly the protein that you have in silent flagella. And what we did was cultivating the model system that I showed you before, this algae, and we cut this flagella. We use the flagella to purify these proteins together with this docking complex. That is a complex that is used to attach the motor proteins to the microtubules inside the cilia and the flagella. Then we have the motors, and then also we use small pieces of this flagella as a nucleator for the microtubules, because you need the microtubules all in the same polarity, otherwise doesn't work. Or then you have these nucleators that are called seeds, and then you can polymerize your microtubules, and they are at a distance that is favorable for the motors to create the interactions. In this way, then you observe that you can it is possible by using biological building blocks. The reconstitution of a beading structure is possible. Of course, you don't see the typical bending, because what is missing here is this cross-linking proteins that I show you at the beginning, and of course, I mean, we need to work on it. Although I mean, it's a challenge. However, if we want to have a schematic representation of these structures, you can see here how it works. You have a patch of motors that then they move toward the joint end, and because again of the elastic stress of the microtubules, they begin to detach from the adjacent microtubules up to the point when then they go back to the space position, and they begin the cycle again. If we measure the distance between these microtubules, then we can characterize this cycle as a function of this distance, and we see that then it goes between open and closed states, and then this state have a different duration from 3 to 30 seconds, and they are characterized by a transition between these two states that it takes something like hundreds milliseconds, and this is can be a hint for the collaborative work of these patches of motors. The problem is that I don't know where is my. No, I'm not. Ah.
It's on your own.
Yeah, we need that one. So if they have a problem, they should.
What should I do? Don't change.
It's transmitting.
Yeah, okay.
Yeah.
yeah, what I was saying, yes, the transition, the collaboration of the models. And then, I mean, by considering this backling, we could have a rough idea of the bending energy that gives a hint about the fact that the structure is driven by the activity of the motors. Now, I told you that we have a patch of motors between this filament. And actually, the reason was because from the image that you saw before, of course, I mean, you cannot really observe such a small details. What we did was I mean, taking microtubules and mixing with proteins at different concentrations. And then what we first saw was that they organized these models organized in arrays that have the same periodicity of 24 nanometers that you find in real cilia or flagella. The reason for this is because we extract the models together with, I told you, with the docking complex. And this docking complex is 24 nanometers long. And actually, normally in the cells, this docking complex and the proteins are transported from the cytoplasm to the cilia or flagella separately. They don't go together. That's why the docking complex, let's say, create and bind on the microtubules in an end-to-end fashion, and then the motors attaching this on this docking complex. The reason for this, so this paper, the conjecture of this paper is that the fact that it's in this way is because you have that this kind of periodic arrange that then is used inside the flagella for another periodicity of 96 nanometers of another kind of motors. And that's why creating holes in this arrangement would disrupt the interaction. That's why, I mean, we have this periodicity because of the way how we extract the motors. But in our case, they are already attached. They are not coming in separately. And what we observe here is that when you have this kind of mixture, these motors are arranged in patches between these microtubules, I already told you. But these patches have a kind of characteristic length, because it doesn't matter which concentration of motors you put inside this the mixture before the visualization, they always have a kind of length that are around eight motors. And if you, of course, I mean, if you add more motors, they should go somewhere, right? And what they change in this arrangement is the number of patches that you have between these two microtubules. We don't know why this they arrange in this. There is a kind of characteristic length, but I mean, this is a very interesting aspect that needs to be investigated further. What could be, maybe one of the reasons why they are arranging this in this way is, or better, I mean, why they have this kind of array arrangement is the fact that in the group of Joe Howard, they found that when you have the dyname, they interact in the array in a way that one activates the follow-up. That's why in this way, it is easy to understand why we don't find a single motors and always we find an array. Still, we cannot explain why these eight motors length in these patches, and this is, I mean, a very interesting point. Here, I mean, I close this part of the let's say synthetic cilia and flagella, where I show you two systems. and one a little bit like a toy system, and the other one I show you in which way we are trying to let's say to go closer to the biology.
Second one, does the two microtubules remain in the same plane, or is it out of the plane?
Sorry?
The two microtubules were out of the same plane, or do they remain in the same plane?
Allora, so of course, I mean, I think that if you see the movies, you see some movies when they go out of plane, because actually, if you observe, you can see that you have the feeling that then they are in this way, and then they like rotate on the top of each other. The problem is that you are, I mean, for these details, you need another kind of microscopy technique. But I mean, this is, I mean, you don't see when the structure go out of plane because of the velocity that I'm playing. Otherwise, you see how sometimes. No, what I'm saying is that the reason why they're doing this way could be because of this. But this is all only based on some speculation, because with this kind of pictures, we don't have these details.
In the EM, do you see the clusters of 8 always between two microtubules?
Yes.
Or on a single one?
Oh, what do you mean?
They can also bind on a single microtubule, right?
Yes.
And so you always find them when they're between two microtubules, or also when they're separate from each other?
I mean, you can. They also arrange on a single microtubule,
But
of course. But I mean, the thing is, but the idea,
necessary to have a second one to get this cluster size.
because I mean, because the thing is that if you put some ATP in this mixture, then you activate also I mean this domain that bring them to attach to other microtubules. And the idea was, let's see what happened exactly in our configurations. Because if you see, when you see single microtubules, you see that they are decorated in a different way. For example, they have patches in different directions. There is many papers also on this. And the thing is that we wanted to see in which way we have this kind of things that then can bring to our setup, the one that we have that is active.
It looks like in this image the patches on the outside of one of the microtubules. It's not between them.
This one?
Yeah, the yellow, the one that we.
No, I mean, no, I think that. Do you mean that this one looks like? They are like always the modulus between the microtubules, yeah. And at the end, I mean, they really did. I mean, every concentration has something like 100 different, let's say, pairs, and that's why. Going back to this initial cartoon, I told you that the second system that I worked on was active networks that are based on building blocks from the microtubules. And also this project had a kind of biology motivations, because we saw that, in nature, we see that microtubules arrange in a different way according to the function that they have to accomplish. Here we see the spindle in the cell division. Here, for example, the parallel arrangement in the neural cells. And actually, the best example that I found and that I really like is this cytoplasmic streaming that you have inside this Drosophila oocyte, and the change during the developmental stage. You go from a mixing, or the production of a mixing fluid in the early developmental stage, and in a kind of a coherent flow when you are in a late developmental stage. Of course, this fluid and this transition are motivated by the functions. I mean, it's when you are at this level, all the compounds that come inside the oocyte should be equally distributed everywhere, and then makes sense that you have a mixing flow. But here in the late stage, you are in this stage when you need the gradient, as we have seen last week, and then you see this coherent flow. Now, I mean, I'm interested in the transition between these two regimes, and of course, I mean, I don't have the answer yet, but I would like really to work in this with this organism. But the way how we try to see this transition or this arrangement of the microtubules was by using the same system that you know here. This is the setup of the Dogic group, and then we saw also last week. We saw this here, the same models arranged in the same way, and then you use this crowding agent to arrange the microtubules in long bundles. Of course, we changed the setup for our needs by changing the density of the model, so the length of the microtubules, and then we put this mixture inside the 3D channel to let this stuff evolve in 3D. Of course, you know this mixture that this is the result when you use this mixture. But then you have a setup that is confining this structure in a 2D. But I told you that we have a kind of 3D structure, and this is what we observe when we confine this mixture in this volume. You see a contraction of this network along the y direction, and actually you see also in the z. But this is just a top view of a movie from the microscope, and that's why you cannot appreciate this contraction in this direction. And then you can see how long the bundles are. They're oriented along the longitudinal axis of this channel, and they show an aligned arrangement. And then, of course, the emergence of this regular pattern that is characterized by a wavelength. Just because we don't have let's say the possibility with the microscope to do the same things, here we have a kind of schematic representation to show you what happened in this sample. We first have the contraction of this system in a layer, and then you have the buckling in the third dimension. Now, if you just to show you that this is really a waving structure, we can scan this system along the height. You see that these two crests are in focus that, for example, correspond, let me say, to this. And then when you change the focal plane, then you see that you focalize on the other two structure, and then you see that this is kind of wavy structure. It is very interesting if you observe that the transition, let's say, the transition between the crest and the throat can be differently sharp. For example, if you see in this other example, you see that actually the structure presents a very sharp transition between the two let's say the two crests. Quite so different from this that was a little bit more smooth. Other groups show the same behavior, same buckling, the arrangement of the microtubules in a nematic way, and the same contraction of the structure in a layer. But they claim that the contraction is passive, just given by the fact that you have a crowding agent. We use polyethylene glycol. They use pluronic are a little bit different because the pluronics co-block copolymer, but the effect should be the same. However, we, in our case, we demonstrate that in the contraction is active. Because if you just let the system with PEG, but without motors, you see, of course, a small contraction and then the formation of these bundles. But actually, if you remove the PEG and you have only motors, you also have the contraction. The only thing is that you need to wait longer, and that you lose then, let's say, a so visible arrangement as in the case of the PEG. And actually, just motivated exactly by this effect, we tried to compare the contraction effect that you have in active networks and not passive when you use PEG or when you use another kind of crowding agent. And what we realized that actually when you use a pluronic, the contraction was strong, and you were observing some tearing in this network. And we believe that the reason is because the PEG forms these bundles. If you then polymerize them and then you dilute, they form these thick and long bundles. In the case of the pluronic, at least for the concentration that we use, you get a kind of isotropic network. These bundles are thinner, and we believe that the organization of the microtubules in the network then determine the stronger, let's say, transfer of the force of the motors.
Did you try ADP and ABPNP?
No.
Because then you would have motors, but no activity. Would also be
No.
okay.
Just to see, but the thing is that we okay, because you see, because then you cross-link them.
Cross-link you could also do it.
No, no, we didn't. The only thing that we did was mixing them, but without giving ATP. That because considering that at the end, at least, before doing this experiment, I spent quite a lot of time to check what happened when you have motors and you don't have ATP, because at the end you need that they bind to the microtubules, because if they, and that's why, we did, I found that the microtubule binding domain works also without ATP, and if you think at the end,
they don't last, they're not stable without ATP or ATP.
Yes, or either or. Yes, but but they are not. It's not. For 4 or 5 hours, I think that is really.
Well, I guess you.
Okay, very good. But then, if going back to our buckling, layer. This is, after the buckling instability, we observe that the system moved toward a more disordered state. The pattern is not there anymore. You still have bundles, but they are shorter. In order to understand, how, or better, in order to have access to some parameters that are not really directly accessible with the experiment, again, Ramin and his group helped me with this simulation, and that they did with the cytosim, and this is the side view, this is the top view, and then you see that by using the ingredient that we claim that are necessary for observing this phenomenon, you see that the system is able to contract in one layer, show you buckling behavior, and then actually is even able to show you this disordered state with the disappearing of the pattern that I show you during the experiment. At this point, we could have an idea of the forces inside this system during the evolution, and then you see I mean that how this the active stress change, and then we thought that for changing and tuning the wavelengths of this system, that we wanted to act on the active stress. And the first idea that came to our mind was changing the ATP in a range that was 2 orders of magnitude big. But the thing was that actually you could not really find a difference between the wavelengths found for different ATP concentration that could be statistically relevant. The only let's say relation that you can find is the fact that you have system buckling faster, system buckling slower, and the wavelengths of the system is buckling faster is shorter compared to the other one. But this is, I mean, is quite let's say intuitive, considering that the one that is buckling faster has more force, and that's why the wavelengths can be short. To understand, I mean, to give our interpretation of this difference in the results at different ATP, we tried to use the simulation again to see the forces between the anti-parallel microtubules that actually are important for the formation of this for the emergence of this instability. And then we found that actually their value is closer to the stall force of the kinesin. And then one speculation could be that the reason why we don't observe this big change when you change the ATP is because of the stall regime that the models are working. But again, this is at the level of the speculation. Also, because if you observe other active pneumatic systems that were observed tested or I mean analyzed in the last years. This one is from the group of Sages. You see, this, for example, it was in a circular setup, and you see that the pneumatic order is radially, and then you observe this in plane instability, and you see that actually the wavelength of the system is dependent on the concentration of the ATP. Still, I mean, we have this example, we have our example, and I mean, more than my conjecture that I told you at the moment, at the state of the experiment, I cannot, I cannot say further. And but this experiment that I showed you, this is, I mean, the way how they did the experiment is a 2D system, and then you have this kind of circular system, and then you inject the mixture in the middle, and then distribute in a radial way, and is confined at the oil interface. If we try, let's say, a similar setup by injecting our mixture from the center, and then we wait a bit of time, you can see that exactly as in the example of the chamber, the pneumatic order follow the confinement that the shape of the confinement that you have. And these instabilities that again are also this one are 3D, you can see that more or less also in this case, far away from the let's say from the transition of this this geometrical transition, they still let's say emerge perpendicular to the wall of the confinement. I think that this what happened. I mean, what I concentrate on now is what happened in this situation. What happened to these instabilities when you change somehow the impedance or an abrupt changing in the geometry. And with this, I arrived at the conclusion. But don't be too happy because it's not the end of the presentation. I mean, I show you this 3D active nematic that can show this transition from the 3D pattern to the disordered state. As Cristina Marchetti said last week, of course, I mean, we cannot claim that 100% we can have a link with the biology, but maybe can be used to tune a bit the setup and having more information. I show you this, how we tried to modify the wavelengths that is insensitive to the ATP concentration. Of course, I mean, then to modify it, we change the concentration of the motors, but this is, I mean, is more obvious than when you reduce the number of the actuators, you change the wavelengths. And yeah, and then the different geometries can be explored, as I showed you. And as an absolutely last topic that I will show you is how I also want to use these active networks. And the thing is that until now I show you only hard confinement, PDMS chamber with glass, but of course, I mean, they can be confined inside a vesicle, and we have seen studies when you know that they can collapse on the internal shell of this vesicle, but actually if you use other kind of vesicles, they can just contract and showing other behavior like this spinning that you can see in this. This was just a small part that we contribute to a collaborator of ours that needed a contribution of let's say of an in vitro system, but this is very promising because it is easy to link it to the biology. And another way to use a soft environment was this one. This is absolutely the last work that we have. You have always the same network, but you confine in an evaporating droplet. Then you know that you have an evaporating droplet. Of course, you have the flow field that is generated by the evaporation, and we wanted to see how it couples with the activity of this network. You see that you observe this kind of behavior. You see that actually already at the beginning of the experiment, the process already began. And you see this radial, this annulus, where the bundles of the microtubules are arranged radially, and then the system contract up to a point when it cannot contract anymore, and we believe that the reason is that the microtubule network is actually giving a kind of resistance to this pushing effect. And then, I mean, even at the last state when this structure is contracting, is expanding because is losing the activity. In the middle, the system is still contracting. What is happening in this process? So you have the peg that you need for doing your microtubules bundles, and of course the peg works as a surfactant. And for this reason, you have the generation of a Marangoni flow. That's why you have the Marangoni flow that is going inwards. You have the capillary flow that because of the evaporation, and then you have this kind of vortex that is, vortices that are forming inside of this evaporating droplet. And then, I mean, here we show, I mean, the measurements of this two flow that we did with the mixture without the microtubules. And of course, then you see the contraction, and we believe that this network that we already saw is, I mean, is a contracting and also an extensile network, and maybe at least this is the speculation that we have. This is the reason why there is a limit for this contraction, up to the point where, of course, the water is evaporated, and you have the salt increase, the PEG increase that reduce the activity of the network, and of course also the Marangoni flow is reduced because you don't have the gradient in the surface tension anymore. But of course, the thing is that this it could be that you have this behavior even without activity, and for this reason, we first remove the Marangoni flow, and then you see that this structure is only subject to the capillary flow during the evaporation. But actually, you see that the network is still contracting. Now, the amount of contraction is much weaker compared to the example before, because here the fact that you don't have the PEG, you have loose microtubules, and that's why they don't have this favorable distance that give them the possibility to transport this force. But the best example you can see if you remove the activity of the motors. When you have this kind of or better, when you don't have the activity of the motors, you see this very peculiar behavior. You see that in the movement, let's say during the evaporation, the network of the microtubules that are arranged in this way because you have the PEG is not. It has no synergy anymore with the aqueous phase, and then you have this separation. These experiments were done exactly in the same condition as the other one. The only difference is that you don't have ATP, and I hope that in these minutes they don't. Otherwise, I mean, I will try with in the other way that you suggest. But I mean, this is show exactly what the Marangoni flow can do to this system, and actually that you need the activity of the motors to win, let's say, against this reaction with the surface. And with this, I really finished all the experiment that I wanted to show you. At the single filament level, I wanted to show you some beating structure and the possibility to go closer to the biological structure. At the multi filament level, I show you these different systems, and in which way we can use them to learn I mean, some processes in the nature. I mean, with this, I mean, here the people that work with me and that did the work, and they really I'm really thankful. Here all the collaborators, and especially, I mean, Ramin and Andre that helped me with in all the projects that I show you. I collaborate with them, and then they help me with the theoretical description of the processes. And of course, thank you for your attention.
Isabella, lots of cool active matter. I guess now you can do a deer brain microtubules. A question? Yeah. No problem.
In the evaporating drops, how do you know that the amount will flow is due to the bag and not to the temperature gradient into evaporation?
Okay, because I mean, we did, because I mean, we have, so if you do the experiment in a chamber where you have the controlled temperature, but you don't control the humidity, then you have, I mean, you have this effect, right? I mean, you can control the temperature. Of course, let's say you have two things. First, we consider that we have experimental rooms that have a constant temperature, but of course, I mean, you cannot rely on such a big room. But then on the top of this, you have a kind of controller for the temperature. Because of course, I mean,
So the evaporation is louder at the edge and
yes,
take out the latent heat.
okay. I see.
There's one of the experiments of coffee stain effect, where the temperature gradients appear in this.
I mean, I should say, I mean, we did all the experiments, let's say, in with fluid without microtubules, like the usual evaporation, so sessile droplets experiment that you do. And for this reason, we work with the Stefan Kapiska, that, by the way, is not in this list. I forgot him. And and this is, I mean, because I should say, I'm before this project, I was not really, let's say, an expert. Not that I'm now an expert in marine flow. That's why I work with Stefan that is, that works with this kind of systems to have his advice, and then he suggested all the control experiments that we could do to avoid, or better, to say, okay, here we eliminate the Marangoni flow. Here we have the capillary flow that is bigger than the other one. And then also what we did was also that I didn't show in these things. We functionalized the surface, because of course you always have proteins, and that's why you have the problem that everything sticks on the surface and you don't see anything. And exactly for this reason, we analyzed, for example, the coffee stain effect on this surface, and you see that if the surface is not functionalized, you have the classic ring, the one that we know, because all the structure goes on the edges. But in the case of the normal of the functionalized surface, you have a kind of a gap between this. That's why I think that the system works, because we try as much as we can to keep the temperature constant. We have the peg, and we have the surface that is functional. If you remove one of these things, of course you will get the same pattern. And if you have all of them, but you will have the motors.
It would be interesting to try it. If you can somehow measure the temperature locally, it's not so easy.
So no.
Or put it on a sapphire or so, where the conductivity of the substrate is much larger.
Yeah, this is an idea, because I mean now this is I mean we are working on because it's a very easy setup, and this is quite interesting. This actually, I should say, I found very interesting also the behavior of the passive network when you don't have the motors. But this is, I mean, this is not active matter anymore, but this is still very, very interesting.
More questions? Oh yeah, go ahead.
So I mean, let's forget about the evaporating ground.
Yes.
Let's look at what happens inside the vesicle.
Yes.
So you have any, apart from those spinning things, what is I guess more of my question, what is the interaction between the filaments and the vesicle surface? Is it just like it's
At the moment,
collision, or
in the I should say, in this project that we had, we could not see an interaction, because you see that the network was contracting and the rest was just intact. Exactly for this reason, because I was waiting some interaction between this with the membrane. Now, I mean, what we are planning to do is connecting them if you can see that force transfer with this membrane. Of course, I mean, always with the idea that we need a kind of system close to biology, and that's why just, I mean, to use the active network to deform confinement.
And have people looked at the shapes of these vesicles, for example, as you increase the concentration of your, the filaments and things inside?
At the moment, I should say, I mean, all the studies that I know at the moment, either they put some stuff inside, but it's mainly used to see if you can create a kind of synthetic cytoskeleton, and otherwise, I mean, we have the work, for example, it's 10 years ago, I think, where they let this active network, let's say, adhering on the surface or on the internal surface of a vesicle, but this was a little bit different because this is a lipid double layer, and the other one was a vesicle. But I mean, mainly this. I mean, now as this one is not a vesicle that is inside the oil, this is easier to deform. This is a little bit more complicated, and you need, I believe, an anchor system to deform it. And this is the reason, for example, now there is all these people that are trying to replicate the cell division, and of course they need something that knows exactly where it should go, and then contract and deform these things. But at the moment, is
Wasn't it Deborah Falkenson or somebody, or was it Zachman's lab also, where they showed that the microtubules would push the membrane out in
Yeah, yeah.
vesicle?
yeah, these are yeah, exactly. I think
Deborah.
that I think someone from a Bausch and
No, that was active. Oh, wait. I think-
some microtubules.
yeah, but I remember seeing the stuff that really pushes out
Yeah, they are
it was vesicles.
watering oil.
No, not emotion. That was a vesicle.
Really?
Pretty sure. But yours was also vesicles, right? Not emotions.
This one, yes.
Vesicle, okay. So maybe it's the length or stabilization, or there was Taxol involved, or I don't know.
Well, I mean, I should say, I mean, I always, I didn't know that it
The
was.
vesicles are pretty soft, so you would think that it can easily deform them.
Yeah, I mean, but still, I mean, you need, if you have this kind of structure, you need a strong force to deform them. That's why, I mean, I think that linking them would be a good way to deform this confinement, this compartment confinement.
I was wondering, in the first experiment you showed with the overlapping microtubules in the buckling, can't you just look at fluctuations and sort of look for a fixed point in the fluctuations to identify where the motors are? Because they're doing this, but they pivot around the point where the motor is. So if you quantify the fluctuations along the filament, you should find fixed points.
Okay. Yeah. No, I didn't do it.
So I guess it was pretty noisy. The image was noisy, so maximum intensity might be tough. But if you track the backbone and then sort of superimpose the traces,
Okay.
might be doable at home.
Yeah. I mean, you mean in the two?
Yeah, when you had two and they were buckling.
Yeah, yeah.
It's a classical, you always see that when you have motors on the surface, they buckle.
Yeah.
But in the extreme case, when
yeah. Okay. Yeah. No.
you get nodal point pivoting. That was
Okay.
the
Yeah. I
Audi
can.
experiment. So the motor is fixed, so they should
Yeah.
pivot around a fixed point.
Yeah, but this is also the question. Is the model fixed? Because I mean, the idea. I mean,
More or less.
originally, the reason why I functionalize the surface is to avoid that you have that the model is always fixed in the same position. Then, of course, I realized that without having any labeling, you are not able to verify if it's true or not. But this one could be a way.
Any more questions? I guess we can postpone the rest to individual discussions and have coffee anyway. Thanks again, Isabella.
Thank you.